You are welcome to today's facilitation period, and as usual, our time for facilitation is 4 to 5 p.m. every Tuesday. So, and what we are going to consider this time around is Unit 4 of our course materials, which has to do with the kingdom fungi. So it is the kingdom fungi we are going to discuss during this facilitation. And when we talk about fungi, it's one of the six classification kingdoms of living things that we know. And we have been talking about the six classification of living organisms, which we all know since the beginning of this facilitation period. And then one of the major characteristics of the kingdom fungi is that they do not have green pigment that enable them to carry out photosynthesis. And what that means is that they lack chlorophyll in their cell. So, and then they are all eukaryotic. They are more advanced than the prokaryotic that we had discussed in the past. So they are also multicellular, except very few of the members that are unicellular, such as the yeast. So fungi usually secrete enzyme which can disintegrate or dissolve almost all materials. That is, any substrate they come in contact with, they secrete enzymes that may dissolve it or degrade it. And they also know that their body, except the unicellular members, which we just mentioned, the yeast, is made up of delicate thread-like structure called hyphae. So it is this hyphae that radiate that penetrate the substrate on which the fungi grow. So the hyphae is collectively called mycelium. The plural of mycelium is mycelia. And the wall of the hyphae may be made of chitin and not cellulose, as found in a plant cell. Then what are the diagnostic features of the member of the kingdom fungi? So since we said they are eukaryotic, they are multicellular, except those that are unicellular. And those that are unicellular do not have hyphae or mycelium. And the main body of fungi is made up of hyphae, which we have said are long thread-like structures. And all are heterotrophs, since they do not contain the green pigment that will enable them to carry out photosynthesis. So they rely on the food manufactured by other organisms. So they lack chloroplast, which contain the chlorophyll for photosynthesis. So, and what that means is that the members of this kingdom cannot carry out photosynthesis. They are all saprophyte. That is, they are saprophyte. Some of them are saprophyte. Some are parasitic in nature, and they usually absorb soluble organic substances from the material around them or the substrate in which they grow. So, looking at the structure of fungi, we have said they are eukaryotic and they have membrane-bound organelles, unlike the prokaryotic organisms. So, this group have membrane-bound organelles, and they lack plastid, which contain the green pigment for photosynthesis. They have no chlorophyll present in their cells, and the hyphae found in fungi are either cynocytic or they are septate. So the cynocytic one is that the hyphae that do not have crosswall. So their cell wall are usually rigid, and then we said they contain chitin. So this chitin will be layered with mannans and glucans. That is the nature of the cell wall, the chitin that made up the cell wall of the fungi. So it could also contain other polysaccharides. That is, the chitin may also contain other polysaccharides in association with polypeptides. And in some primitive type of fungi, that is the lower fungi, the cell wall may contain cellulose. And then the inner to the cell wall is the plasma membrane. That is the one that you find immediately after the cell wall. And then the plasma membrane is a typical bilayer membrane in addition to the presence of steroid in them. So the fungal membrane possesses what we call ergosterol in contrast to cholesterol found in animal cells. And there are various organelles that are found in the cytoplasm. So such organelles that are found in the cytoplasm of fungi include the mitochondria, the Golgi apparatus, the ribosomes, the endoplasmic reticulum, then even others include the reticulum and the endoplasmic reticulum, the lysosomes, the microtubules, and the membrane enclosed nucleus. So there is an interesting and unique property of a nuclear membrane in fungi, and this unique and interesting property, the membrane persists throughout the metaphase of mitosis. That is one of the uniqueness of the nuclear membrane in fungi. So, and this is unlike in plant and animal cell, whereby during this mitotic cell division, the membrane will dissolve and reform later. So, the nucleus in fungi possesses pair chromosomes that are found in them. Then, how do they look like? That is the physical appearance of fungi, the morphology of fungi. They can appear in two forms. As we have said before, those that are filamentous, they have hypha. Then those that are not filamentous, they do not have thread-like structure like yeast. So, but all fungi have a typical charotic structures. So then we can now classify them. And how do we classify fungi into the various groups? So, and then basically we have 4 phyla. The singular is phylum. So, but 4 phyla are found in the kingdom fungi. And the 4 phyla that are found in them on the basis of the classification are the Ascomycota, the Deuteromycota, the Zygomycota, and the Basidiomycota. So these are the 4 phyla that are found in the kingdom fungi. Then we are going to look at each phylum. So, and look at their characteristic features and what can be used to recognize them as a group. So the Ascomycota show the following characteristics. They are single cell. The mycelium is septate. The conidial formation is common. The ascus usually contain 8 ascospores that are formed endogenously. That is, they are formed inside. Then the sexual reproduction is reduced to fusion of 2 compatible nuclei. So they have motile cells and they lack motile cells. That is, the motile cells are absent in the group. The ascoscarp is multicellular and bear the asci. Then the ascoscarp that is open and look like cup or saucer is called apothecium. And then when the ascoscarp is over or they appear like flag-shaped with a small apical opening, it is called perithecium. So if if the ascoscarp is completely closed, it is called cleitotecium. So examples of Ascomycota and Saccharomyces, which is the group of yeast. We have the Aspergillus and we have the Penicillium. They are the typical members of the Ascomycota. So this is a structure of yeast, and you can see like a budding coming out from the top. That is a small outgrowth in them. So you see those that do not have that small outgrowth. This is a typical structure of yeast as observed under the electron microscope. Then the phylum Deuteromycota, and this is an interesting phylum. So the they have a specific features that distinguish this phylum from other group, and we refer to them as the group that contain fungi imperfecti. That is, those fungi that their sexual stage has not been identified. So they have diagnostic features, and the diagnostic feature that recognize this particular phylum is that the mycelium is septate. They have a cross-wall mycelium. So the sexual or perfect stage is unknown, as I've said. That is, the sexual stage is not yet recognized. And their reproduction is mostly by means of conidia. They form conidia. And a typical example of this phylum is Fusarium. Fusarium is a parasite, one of the parasitic fungus that cause diseases in plants. Then the phylum Zygomycota, their own diagnostic feature include that the mycelium is unseptate and sinusitic. That is, the mycelium have no cross wall. The sporangia has innumerable sporangiospores. That is, there are many spores that are found in the sporangios and spores. And then the sexual reproduction is eugamous, and this is found in the member called Oomycetes. The sexual reproduction is isogamous in the group called Zygomycetes. Then there are some that are biocelate, and they are motile cells, and they are produced by many species of this phylum. So the zygote is unicellular and simple in nature. And some of the examples of Zygomycota are the mucor, the cystopus, and the rhizopus. So they are the examples of this phylum. The sexual reproduction is isogamous in Zygomycetes. So, and as I've said, the biocelate motile cells are produced by many species, and then the zygote is unicellular and simple in nature. So we have mucor, cystopus, and rhizopus. This is the structure of rhizopus. As you can see, the spore. We have the sporangiospores that scatter. See the sporangium, the apophysis. We have the columina after the spores might have been discharged. So we have the sporangiospore, on top of which we have the sporangium. Then we have the rhizoid. Rhizoid are the one that penetrate into the substrate. Then the phylum Basidiomycota. Here we found the higher mushroom, those that are highly developed. And they have their own characteristic features also, which include the septate mycelium. Then their conidium formation is not a common feature as we have seen in other phylum. So the basidium usually has 4 basidospores that are formed exogenously. In this group, their basidospores are formed exogenously, that is outside of the cell. And then the sexual reproduction is reduced into 2 compatible nucleus, or fusion of 2 compatible nucleus. So this fusion of 2 gametes is called karyogamy. That is, when the 2 gametes fuse together, the process is called karyogamy, and that is the union of the 2 nuclei of the 2 gametes. So, and this fusion is in the young basidium. It takes place in the young basidium of the cell, and then the motile cells are absent in the group. The basidocarp, that is the folding body, is unicellular and highly complex. So the basidocarp bear the basidia, and it is often open. Sometimes it may close. There is what we call clamp connection, which is common in this Basidiomycota. So the examples of Basidiomycota are Pusinia, Pustilago, and then we have Agaricus, and that is the mushroom. So we have some members that are parasitic, and then we have mushroom as the member of this Basidiomycota. This is Agaricus, is a mushroom. So this is a picture of a mushroom, which is a member of the Basidiomycota, the mushroom. Then what are the adaptive features of fungi? That is, what are the structures that enable them to survive or to live successfully in their environment or where they found themselves. So, and some of these adaptive features include that they are mostly found in damp environment. That is, where the environment is wet, is where you find the fungi. So they grow best in damp habitat, and they are found wherever organic matter is present. So anywhere you see fungi, it means there is presence of organic matter. Otherwise, they cannot just sustain themselves where there's an absence of organic matter. So they are a successful group of land organisms. So they possess several features in their body and reproduction that enable them to survive successfully in their habitat or in their environment. So it is the feature that adapt them to terrestrial mode of life. One of the major difficulties of organism is ability to live successfully on land. So but there are some of these organisms that have some features that enable them to live successfully on land. So and some of those features include fast spreading hyphae in fungi. It's one of the structures that enable them to survive successfully in their environment. So their cell wall is chitinous in nature, which is one of the structure that enable them to survive or live successfully. And then the chitin in their thickened fiber wall is more resistant to decay than cellulose and lignin that is found in the plant cell wall. So they have chitin, and it's one of the unique features of fungi that enable them to live successfully, unlike in the case of plant cell where they have cellulose, which can easily be degraded. Then they have rhizoid. As we have seen rhizoid in the case of rhizopus, and it is this rhizoid that anchor the fungus to the substrate and also serve as an organ of nutrition because it absorbs and digests food for the fungi. So they do not have organs of locomotion, no flagellated cells in them. So they lack flagellated cells throughout their life. So then one of the main features that enable them to survive also is production of numerous spores. So they produce large number of small non-motile spores that are scattered and can be carried away from the parent organism into different environment, and that's why you find fungi almost everywhere. So the spores are called conidia with thick protective covering in them. They can tolerate temperature that is very high. There are some of them that can even live a temperature below the ice that is below 0 degree centigrade. So below, if as low as -5 degrees centigrade, which is below freezing point. Then some of them can live in a very high temperature above 50 degrees centigrade. So, and that's why even those that are pathogenic can live. Ordinarily, the ideal temperature for fungi to survive is between 27 and 30 degrees centigrade. But you can discover that some of them will live successfully at as high as 50 degrees centigrade. So these are the kingdom fungi. We have looked at the various groups. We have seen their characteristics. We have seen what enabled them to survive in their habitat and environment. So let us just briefly look at the activities that you can carry out on your own for this group. However, some of these activities will be demonstrated during the practical class that is being organized by the Faculty of Sciences. So, one, if you really want to observe a fungi or a fungus, what you need to do is that you place your wet bread or damp bread or even eba or aku in a warm and moist place and leave it for about 3 days. Then you will observe a certain color after that 3 days on the food. Then you should guess what those colors represent. That is one activity. I want you to do. It is something you can practicalize on yourself. Then, secondly, you can go around, look for old or decaying logs, any wood around your houses, particularly those of us that stay in the villages. Then, observe critically whether you can see any white or creamy plant-like growth around or near or on those decaying logs of wood. Then, what do you think they are? So these are some of the activities I want you to carry out on your own. That is a practical exercise so that you can demonstrate on your own. So that is, we come to the end of the kingdom fungi. So we will now enter into another kingdom, which is the plant kingdom, which is the highest group, one of the highest kingdoms we have. Apart from animalia, the next higher group of organisms are the plant kingdom. So, and under this plant kingdom, there are some objectives. That is, what are the intended learning outcomes that we want the student to know. So, at the end of this discussion on the plant kingdom, so the student should be able to know the division in the plant kingdom. Which group are the plant kingdom divided into? Then the various habitat of the division of the various members of the plant kingdom. Then the students should be able to identify members of the division. So what are the members that make up a particular division of the plant kingdom? And then what adaptations did they exhibit by each of the division? So there should be some characteristic features that enable them to live in their various habitat where they are found. Then the plant kingdom generally is divided into two major groups. And the two major groups are those that produce seeds and those that do not produce seed. Those that do not produce seed, we refer to them as seedless plants, while those that can produce seeds are called the seed plants. So the seedless plant have two divisions too. The division bryophyta and the division pteridophyta. These are the two divisions of the seedless plant. And these two groups, they do not produce seeds at any point in time of their life cycle. The seed plant is also divided into two groups. We have the gymnosperms, in which their seeds are naked and not produced in any enclosure. And then we have the second group also, the angiosperms. The angiosperms have their seed produced in an enclosed structure called the ovary. So, and then they are the group of plant kingdom that produce seeds. So, we are going to look at the division one after the other also. So, the division bryophyta, they are the first division in the plant kingdom. So, and they are the earliest land plants. They form a transition between terrestrial and aquatic plants. In fact, we refer to them as amphibian plants because they can live in water and they can also live on land. So unlike in the case of algae, they are all multicellular and they are very complex in nature. Then what are the features of the division bryophytes or bryophyta? They are highly primitive and they can only survive where the environment is damp or shady places. That's where you find members of bryophytes or bryophyta. And they do not have true roots, stems, or leaves, as we found in the higher plants. So they lack root, they lack stems, and they lack leaves. So they do not have the vascular tissue of xylem and phloem. Of course, we know the function of the vascular tissues for transportation and absorption of materials in the plant. So they do not have it, and that's why they live directly inside water or where the environment is damp. So because they lack these structures that will be able to absorb and transport water, mineral element, and food in their cells. So Their sizes are very small in nature. The gametophyte generation is the dominant phase of the life cycle of the bryophytes. So, and then the sporophyte is attached to and is independent on the gametophyte, and is dependent on the gametophyte for its nutrition and survival. So the spores are produced by the sporophyte of the bryophyte, and the spores are produced in capsules on a slender stalk above the gametophytes. That is where the spores are usually produced. Then how do we classify bryophyte? So that is the classification of the bryophyta. So the division is divided into 3 classes, and the classes are the Hepatica or liverwort. Typical example of this division is Mercantia or Lycia. Then we have another division called Anthocerotae or horn liverworts, and the example is Anthoceros. Then another division is Muzi or Mosses. Example is Funaria. Funaria is common around. We will talk about it as we go on. This is a structure of Mercantia, which is an example of bryophyte. Then in the Mercantia, we have a gamete cups where the spores are produced. So this is a structure of the gamete where the spores are produced. So they look like a cup inside which the spores are formed. Then this is an example of Anthoceros agrestis. So this is how it is look like. So possibly maybe during the practical classes, we might show you some of these plants, bryophytes. Funaria, so hygrometrica is another example of bryophytes. As you can see the structure also here. Then what are the adaptive features of bryophyte? So the bryophyta have pores on their leaves which allow the entry of atmospheric oxygen, that is exchange of gases. And 2, air disperse asexual spores in the bryophyta, and the embryo is developed within the female sex organ. So they cannot withstand desiccation because they do not have a cuticle that protect their cell wall. Excuse me. So the bryophytes are still restricted to moist areas, damp environment, since they lack the xylem and the phloem that could transport materials. So they are usually restricted to moist environment. So they still require aquatic medium for the male gamete to travel to the female egg before fertilization can occur. In fact, their reproduction takes place in aquatic environment. So there must be presence of water before they can carry out reproduction in the bryophyte. So then the next division is the division Pteridophyta. And this is a non-seed vascular plant. This group have vascular plant unlike the bryophyte. So they are plant with xylem and phloem, and their dispersal relies on spores and not seed. They do not produce seed. So the sporophyte is the dominant, unlike in the case of the bryophyte, where it is the gametophyte that is dominant in them. Then what are the features of this division? So they are specialized plant, and their sporophyte is the dominant, as I've said, and very conspicuous during their alternation of generation. That's what we call alternation of generation in these lower plants. So that is one cycle alternates the other regularly. So the gametophyte generation of pteridophyte is small and found in the wet places, and then the sporophyte is differentiated into the stems and what we call leaves. So they have vascular system that is made up of xylem and phloem, and the epidermis of the pteridophyte are covered by cuticle. So that one, and they also possess stomata. The cuticle is the one that prevents desiccation. So from the cell of the pteridophyte, which we said is lacking in the bryophyte. So the sporophyte produces spores, and then the spores could be homosporous, that is, they are of the same type, or they could be of different types, which we refer to as heterosporous. Then the classification of the pteridophyte. So we have 4 classes in the division Pteridophyta, and the 4 classes are: one, we have Psilopsida. Example is Xylotum. This is the structure of the xylotum. That is one of the classes under the division Pteridophyta. Then the second division is Lycopsida, and a typical examples are Lycopodium and Selaginella. So this is a structure of a Lycopodium, as you can see by the right hand side of this slide. So it's a typical example of the Lycopsida. Then the third division is Sphenopsida, and we have 2 examples: the Hostates and Equisetum. You can see the structure of a Equisetum by the right hand side there. So that is a typical example of the class Sphenopsida. Then we have the fourth class, which is the Pteropsida or Felicinae. So example is Fern. Then we have Asola and Adiantum as a typical example of this class. So the structure you can see there is the structure of Fern. So these are the members of the Pteridophyta. Then what enabled them to survive in their various habitats or environment? One, we have already said that they have true roots, leaves, and stems. These are some of the features that enable them to live successfully in their habitat. They have a vascular system that is made up of xylem and phloem, and as I've mentioned before, that these are organs of transportation in the plant tissues. So the xylem is the one that conducts water, while the phloem is the one that conducts food. And then the epidermis is covered by cuticle, and I told you the function of cuticle is to prevent desiccation, particularly the land plants. The land plant have this structure to prevent loss of water in them. Then the epidermis has stomata also, which control loss of water. So, of course, stomata is for exchange of gases and also for elimination of excess water in the plant. So the stomata can close and can open depending on the nature of the plant. If there's excess water, it will open to release the excess, but when there's insufficient water, it will close to prevent loss of water. So that is about this group, and then we are going to look at the activities also. So what I want you to do is that if you are living very close or to a forest, or even in your environment, just go to a damp world, particularly during this rainy season. You will find some greenish tufts growing on the wall, particularly on completed buildings. So, and what you need to do, you just gently collect some and observe under hand lenses if you have. So we will provide it, and then what you will see there now are the members of bryophyte, and a typical example is the Funaria. So they are the green patches that you see on the uncompleted wall when the wall are damp. So what you will collect is the gametophyte of the life cycle of moss plant. So please try to identify this thing. So, and as I've said, we will provide them to you during the practical class if you have opportunity of attending the practicals. Then you should go to a nearby stream or farmland or where you have palm trees growing. There you will see fern that will grow on palm tree or even on the forest floor. Then you can collect some ferns gently along with the rhizome. They have rhizome. The rhizome is more or less like substratum, so that attach them to the habitats. So this is the sporophyte generation of ferns. Then you can collect a leaf, and under the leaf, that is the pinna, we call the leaf pinna. So under the leaf, you will see under the surface. What we see is the sori, which are the spores that are responsible for the reproduction.